Hello, my name is Momar Kasse,
My name is Sena Ugur.
And my name is Brian Nguyen.
and we are from Harmony School of Advancement here to present our science fair subject, DNA paternity testing using simulated gel electrophoresis. So here in our introduction, we have our problem statement, which shows that it is difficult to determine the accuracy of paternity tests due to an inaccuracy found in the amount of DNA samples tested. So our purpose of the experiment was to use a module to introduce students to the use of DNA fingerprinting in a hypothetical paternity determination. Our research question: how does the number of genetic markers analyzed in gel electrophoresis impact the accuracy and reliability of paternity testing results? So, our hypothesis was basically if DNA fingerprinting techniques using genetic markers are employed in a simulated paternity experiment, then the experiment will demonstrate a high level of reliability and accuracy determining the biological parents of the simulated child because genetic markers used in DNA fingerprinting are known to exhibit unique variations among individuals, providing a reliable basis for identification and comparison. So we used DNA analysis with fingerprints using PCR, gel electrophoresis, and our experiment will showcase the effectiveness of genetic markers in accurately determining the parental relationship in paternity case.
Variables. Our independent variable was our DNA samples and our different techniques for DNA printing. Our dependent variables was the accuracy or success of determining the biological parents of the child and our percentage of correct parentage determinations. Our control variables included the temperature and duration of PCR amplification consistent, using the same concentration of DNA samples, using the same agarose gel concentration in electrophoresis conditions, and ensuring the same analysis and sizing methods for all of our DNA fragments.
And here we have our research problem. So our experiment aims to familiarize students with DNA fingerprinting techniques through a simulated paternity determination. Students compare the DNA fingerprint of a child with that of his parents using techniques from genetics and molecular biology. The experiment focuses on teaching students about restriction enzymes, PCR, and electrophoresis. Students perform agarose gel electrophoresis to separate DNA molecules of different sizes. They load, run, analyze, and size pre-digested PCR samples to determine the biological parents of the stimulated child tested. It is important to notice that the experiment doesn't really involve human DNA, but it serves as a hands-on learning opportunity for students to understand the principles and procedures involved in DNA fingerprinting and paternity determination.
Next, we have our materials, which include horizontal gel electrophoresis apparatus, the DC power supply, automatic micropipettes with tips, balance, microwave, hot plate or burner, bleach, pipette pump, 250 milliliter flaster beakers, hot gloves, safety goggles, and disposable laboratory gloves, small plastic trays or large weigh boats for gel destaining, DNA visualization system, aka a white light, distilled water, agarose powder, concentrated electrophoresis buffer, quick strip DNA samples, and 10x flash blue stains.
So, in our procedure, after we equip the proper protective gear, we combine 0.6 milliliters of electrophoresis buffer, 29.4 milliliters of distilled water, and 0.24 grams of agarose powder to create our agarose solution. Microwave the solution for a minute, continuously swirling the flask to mix our solution together. While cooling the agarose gel to 60 degrees Celsius, we sealed the ends of the gel casting tray with the rubber end caps. We then poured the cooled agarose solution into the gel casting tray and allowed it to solidify overnight in the fridge. After allowing the gel to solidify, we removed the end caps and combed to reveal the gel. We placed the gel into the electrophoresis chamber and covered it with 400 milliliters of electrophoresis buffer. We then punctured the foil overlay of the quick strip with a pipette tip and loaded the entire 35 microliter samples into the different wells. After loading all the wells, we placed the safety cover on the chamber and made sure to align the gel from the negative electrode, black, to the positive electrode, red. We performed the electrophoresis at 150 volts for 35 minutes for maximum accuracy. After the electrophoresis was complete, we removed the gel and casting tray from the chamber. We then stained the gel using 1 milliliter of 10x flash blue stain and 149 milliliters of distilled water, allowing it to soak overnight. We removed the gel from the staining liquid and visualized the results using a white light visualization system. The DNA appeared as dark blue bands on a light blue background, and finally we cleaned our station.
All right, heading into our safety, we use glove and goggles, which was worn routinely as we practice laboratory safety. We also exercise extreme caution when working with the equipment that is used in conjunction with the heating and melting of reagents. And we also didn't use pipette reagents, we used pipette pumps. And we also exercise caution when using any electrical equipment, as stated by Sena when we were performing gel electrophoresis. And we also wash our hands thoroughly with soap and water after handling all the reagents or biological materials in the laboratory.
So, some of our investigation questions include: why do different individuals, such as siblings, have different restriction enzyme recognition sites? What is the function of PCR primers using DNA paternity analysis? Why is there more than one single locus used in an actual paternity test? And why do we not use probes in this DNA paternity simulation and still obtain results?
Here we have our pictures where we are creating the gel for the experiment, as well as us loading the samples into the wells of the gel, and also staining the gel and putting it under a white light to see the results.
Data and analysis. So here we have our graphs and our actual gel, which shows the lanes that we needed to use and the different molecular weights of all the samples that we use. So basically, as paternage can be determined by a child's DNA profile, we analyze the DNA profiles of the mother and the child and identify DNA fragments. Some fragments for the child were absent from the mother, meaning these polymorphisms were inherited from the biological father. This was explained by comparison and results father number one in the child. So the child's DNA fragments fit both the mother and father number one. The mother cut DNA with the enzyme had ladder sizes of 3,652 and 630, and father 1 DNA cut with the enzyme had 3,652, 1,300, and 700. Therefore, the child DNA cut with enzyme obtained all DNA standard markers of 3,652, 1,300, 700, and 630.
In conclusion, the hypothesis that DNA fingerprinting techniques utilizing genetic markers would demonstrate a high level of reliability and accuracy in simulated paternity determination, which has been validated through the successful execution of the experiment. Our hypothesis was correct. Unique variations exhibited by genetic markers among individuals, coupled with the precision of techniques like restriction enzymes, PCR, and electrophoresis, have proven to be effective in accurately identifying and comparing parental relationships. This confirmation underscores the robustness and credibility of DNA fingerprinting as a powerful tool for paternity determination, offering valuable insights into the biological parentage of individuals.
So here we have some applications and further research. So basically, this could be used for forensic science, medical genetics, and ancestry determination. And for further research, it could be used for population-specific studies, technological advancements. Some ethical considerations could be like, privacy, consent, and the responsible use of genetic information. And it could also be used for validation in larger cohorts to extend the experiment to a larger sample size and diverse demographic groups to further validate the reliability and accuracy of DNA fingerprinting and paternity determination across a broader spectrum of genetic variation. This has been our PBL presentation. Thank you for your time. We hope you enjoy the rest of our PBL.
Signing out. Good night.